Good morning, everyone. Welcome to our session today, From Satellites to eDNA, U.S. Geological Survey Innovations for the Changing World. As you can tell from our title screen, we have a very full agenda today. So before we jump in, just want to say, if you have questions throughout this presentation, please hold them till the very end. There will be time after all of our speakers to present to ask your questions. And without further ado, I'll pass it off to our first speaker to kick us off, Dr. Annalise Bloom, Deputy Assistant Secretary for Water and Science at the U.S. Department of Interior. Deputy Assistant Secretary Bloom, the floor is yours.
Thanks so much, Joe. It's great to be here today. I work with the team at Department of Interior, which has responsibility for the U.S. Geological Survey, or USGS, and the Bureau of Reclamation. And just a quick bit of background about USGS, in case you don't know it. Agency provides reliable scientific information to describe and understand the Earth, minimize loss of life and property from natural disasters, manage water, biological, energy, and mineral resources, and enhance and protect our quality of life. And our Secretary of the Interior, Deb Haaland, has directed us to unleash the science. And so part of this charge is providing decision makers and managers with the best available science to inform decision-making and policy. And part of this is also sharing incredible science coming out of the USGS with the global community. So it's a delight to be part of this event at World Water Week virtually today. As we're all too well aware, the impacts of climate change are exacerbating global water crisis, increasing the frequency of water-related stressors. These challenges threaten food security, stability, and contribute to the loss of biodiversity. But fortunately, there's rapid innovation in research and science that can provide decision makers with sound science to inform better policy, climate adaptation, and mitigation strategies. And as the Department of Interior continues to implement President Biden's Investing in America agenda, the science that USGS advances could not be more important for addressing challenges like the climate crisis and achieving our department's far-reaching goals on behalf of the American public and the world. And today we'll hear from 5 outstanding USGS researchers who will showcase examples of cross-cutting science, technical tools, and information delivery methods that are advancing our understanding of the changing water cycle and climate. So we'll hear first from Dr. Christopher Crawford about the Landsat satellite, which has been measuring Earth's terrestrial, land, and aquatic environments for more than 50 years and provides a yearly economic impact of over $3 billion worldwide. Next, Dr. Merritt Harlan will dive into how the USGS is measuring water using satellites. We'll then hear from Dr. Brian Pellerin about a program which is developing new sensor technologies and providing high-resolution real-time data on water quantity, quality, and use. Dr. Laura Norman will highlight low-tech but highly impactful nature-based solutions and innovations being used in dryland streams. And last but not least, Dr. Catherine Richter will highlight how environmental DNA is opening new frontiers in biodiversity science, accelerating our ability to detect rare species and understand aquatic communities. So these science and tools we hear about today are really only a small glimpse into the wide array of ways that USGS is advancing and applying science to understand and inform decision-making related to climate change and global water challenges. Personally, I'm actually trained as a hydrologist, so really looking forward to hearing from all these scientists today on such a diverse range of innovative and important tools and capabilities they're using to advance our understanding of the global water challenge and climate change. And I also am extra grateful to these folks for condensing their research into a mere 7 minutes, which as a scientist, I know is very challenging. So thanks so much. Without further ado, back to you, Joe.
Thank you so much, Deputy Assistant Secretary Bloom. And with that, we'll pass it off to our first presenter, Dr. Chris Crawford, to discuss Landsat measurements of the global environment. Chris, the floor is yours.
Hey, thank you, Joe, and I'm pleased to be with the Deputy Assistant Secretary for Water and Science today, as well as my USGS colleagues. I'm a research physical scientist at the Earth Observation Resources and Science Center in Sioux Falls, South Dakota. I work with the Landsat project and lead the project science for the USGS. Thank you. Next slide. Landsat is really a joint partnership between NASA and USGS. NASA leads the development of the space segment. That includes instruments, spacecraft launch, on-orbiting commissioning, and checkout. The USGS leads the development of the ground segment. So this is the ground station development, data processing, data products, and data access, and then assumes long-term mission operations throughout the lifetime of the mission. Landsat has been measuring Earth since 1972 in the visible and near infrared wavelengths, and then in 1984, with the advancement of Landsat 4, the mission added shortwave infrared and thermal infrared observations, which continue today. For 2/3 of the lifetime, Landsat has had two observatories imaging Earth 8 days apart for all global land masses and nearshore coastal regions. As is already mentioned, the Landsat record is now 50 plus years, with our current generation of Landsat 8 and Landsat 9 providing high quality active imaging on a daily basis. Next slide. So Landsat has a distributed ground network across the globe with its international partners, and it receives data as the satellite passes over. That data is then transmitted back to USGS Eros in South Dakota for processing, archiving, and distribution using a data archival collections schema. Landsat maintains its instrument and data calibration standards as an international Earth observation reference standard. It processes the raw data using level 1 and level 2 science algorithms to maintain the radiometric and geometric characteristics and data quality. This rigorous processing chain ensures that Landsat can be used as an unbiased environmental and climate data record for quantification of global environmental change, and more importantly, enabled through time series analysis and interpretation. And the short video you're seeing is sort of just a visual representation of that processing chain. Next slide. So the status of the Landsat archive. So the consolidated global Landsat archive is managed by the USGS and provides more than 50 years of systematic global observations. Landsat 9 and Landsat 8 current generation are conducting global surveys on a daily basis, acquiring up to 1,500 images per day from pole to pole. The color-coded geographic patterns that you're seeing here on the screen represent the evolution of Landsat's data acquisition over the past 5 decades. As you can see, early in the mission, there were regional and continental prioritization. And so if you're interested, USGS has developed a suite of online tools to dig into this globally anywhere you please. Next slide, please. So Landsat are processed into collections, and so this ensures that the data is processed for traceability and provenance at pixel scales. This is really critical for detection and attribution of land and water surface variability and change over time. The most recent collection is Landsat Collection 2, is the current processing standard. And with that collection, you not only now can get the calibrated data, but you can also get the level 2 atmospherically corrected data for surface reflectance and surface temperature. And this is really intended to minimize the effects of the atmosphere as the satellites are observing Earth. And this is really a fundamental advancement for the USGS. As already has been mentioned, Landsat is a significant investment for the United States, both at the national and international scales, according to USGS economic analysis. Next slide, please. So one of the advancements or innovations in the spirit of this session is that for a long time, Landsat has been sort of viewed as a terrestrial land mission, but we know that there are water bodies inland and also important coastal regions that surround the land masses. And so in 2013, when we launched Landsat 8, this really unleashed a new era of aquatic sciences. The Landsat data quality from Landsats 1 through 7 was a little less suitable over dark targets because of the degree of noise in the instrumentation, but that's now changed with Landsat 8 and Landsat 9, and so now the radiometric quality on orbit in terms of its daily imaging is really unleashing a new era of aquatic science applications over the past 10 years. Realizing that the USGS has been working with the project to develop advanced processing techniques to generate high-quality aquatic data products that can be used by downstream water quality and limnological applications that desire to monitor the state of U.S. and global water bodies. I think we'll hear from Merit next on what the USGS is doing there. Next slide, please. So data access. Landsat provides free and open data access through a variety of online portals. You can search through the USGS Earth Explorer, you can use our machine-to-machine API programming interface, or you can directly access the data in the cloud. One of the notable advancements for Landsat in its mission history was in 2008, the Department of Interior decided to make the data freely available, and this really unleashed a new era of science and applications. And Landsat is then the most cited scientific data source in the peer-reviewed literature from a remote sensing standpoint. Most recently, I already alluded to that Landsat Collection 2 was processed in recently over the past in the past couple of years. We use the Amazon Web Services commercial cloud, and that's where the authoritative copy of the data is managed by the USGS. And finally, the final slide as I wrap up my comments today. So the Landsat Next mission, the next generation Landsat mission called Landsat Next is really an initial planning and formulation by NASA and the USGS. Landsat Next is envisioned as a quantum leap forward in observational capability with a 3 observatory satellite constellation that will improve the temporal imaging to 6 days globally, increase the spatial resolution of both the shortwave and thermal infrared observations, and expand the spectral sampling in both the V-square and the thermal infrared wavelengths. Landsat Next it is unique in that it's planned to maintain continuity with Landsats 1 through 9, which is an important guiding principle for the mission, is that we must get consistent observations over time to be able to detect the types of environmental and climate changes that we know are important. This concept is also designed to offer synergy with Europe's Sentinel-2 series, which is sort of a sister mission on the European front, and also to support emerging applications such as water quality and aquatics, that's already been mentioned, as well as snow and ice hydrology, which are changing dramatically at high latitude in polar environments, as well as being able to discriminate between some of the key minerals and clays, and to facilitate better understanding of crop residue in terms of food security, in addition to all the terrestrial land applications that we already support today. And so the final slide here is just wanted to highlight that you can find more information by just searching landsat.gov. There's much information, many tools available. If you have specific questions about Landsat data, you can contact Landsat customer service. You can always contact the Landsat communications outreach and lead, or you can contact myself as the representative on the project science. I'd like to acknowledge the USGS Core Science Systems Mission Area, the National Land Imaging Program, the USGS Earth Observation and Science Center, the NASA and USGS project staff, and then finally the USGS NASA Landsat science teams, which have been instrumental in guiding the scientific developments of the mission. And I will pass, that's all I have for you today. Thank you for letting me speak, and I'll pass it to the next speaker. Thank you.
Thank you so much, Dr. Crawford. Next up, we have Dr. Harlan on measuring water everywhere with U.S. satellites.
Thank you, Joe. My name's Merritt Harlan, and I'm in the hydrologic remote sensing branch at USGS, and I'd like to acknowledge and thank my co-authors, Jack Eggleston, Rob Dudley, Tyler King, Meredith Reitz, Gabrielle Sine, and Toby Welburn for the opportunity to speak about their work today. Next slide, please. And as Joe mentioned, today I'll be talking about satellite hydrology and focusing on current USGS monitoring, specifically looking at water quality, including temperature, chlorophyll, and trophic status, as well as water quantity applications focusing on evapotranspiration and river discharge today. And here I've shown an image of the Landsat Next mission that Chris just highlighted, as well as the surface water ocean topography satellite, which recently launched in December. Next slide, please. First, I'll be talking about satellite monitoring of river levels and discharge. For this work, we're using Landsat 8 and 9, as well as Sentinel-2A and B, to look at changes in water extent over time. We're pairing those changes in water extent with changes in water elevation from satellite altimeters such as Jason 3 and Sentinel-6. And we're really excited to start using the SWOT, or surface water ocean topography data, as it simultaneously measures both water extent and elevation in rivers and lakes. In order to look at discharge from this data, we're building ratings between water extent and elevation and pairing that with on-the-ground measurements of river discharge or modeled output of river discharge through different flow law equations to look at discharge. Along with this work, we're planning to increase access to satellite data through developing dashboards. This is a prototype co-developed by the California Water Science Center, in which we are displaying all of the rivers that the SWOT mission is expected to observe in its lifetime. And we're hoping that this dashboard will be a great tool for people to compare satellite observations of water level and discharge to nearby USGS gauge data as well. I'd also like to thank the many international and local partners, both in Alaska and throughout the globe, for their help and assistance. And we're excited to start to expand this work with international partners to collect international river discharge measurements to validate and compare with SWOT. Feel free to reach out to myself or Rob Dudley if you have any questions about this work. Next slide, please. For satellite water quality monitoring, we're using satellite sensors such as Sentinel-2A and B, as well as Landsat 8 and 9 most recently. Here we're focusing on rivers and lakes wider than 100 meters. Our current spatial coverage is limited to the next generation water observing systems basins that Dr. Pellerin will talk about in a few presentations. And these basins include the Delaware, Illinois, Upper Colorado, and Willamette River basins, but future work will expand this coverage to the entire US. Next slide, please. Here we're looking specifically at temperature, chlorophyll, as well as turbidity, and these are from a mix of, so temperature, we're looking at the Landsat sensors and that temperature, and chlorophyll and turbidity, we're using Sentinel-2A and B. For trophic status, we're also using the Landsat sensor, and then for harmful algal blooms, we're using DSIS as well. And here we've shown a depiction of modeled output of the threshold of chlorophyll reaching a certain level where it's then considered toxic. And so we're using satellite data to help show where it might be good to target on the ground future in situ observations of water quality. And so here we're really hoping to pair our dense network of on-the-ground measurements to satellite imagery to build out a robust US-wide monitoring system. Next slide, please. For satellite monitoring of evapotranspiration, we're combining ground-based observations with satellite-based Earth observation to look at evapotranspiration throughout the whole US and combining these measurements with modeled output as well. For evapotranspiration, we're looking at the flux of incoming precipitation and what's leaving through either evaporation or transpiration. And in the US, evapotranspiration accounts for about 70% of the incoming precipitation. And so it's a huge flux in our systems. This map on the top right here shows our on-the-ground flux tower measurements or the ground-based observations I mentioned. And this figure below here shows a Landsat image of Sterling and the modeled output of evapotranspiration. And here we can really start to pick out the crop fields and their importance for modeling evapotranspiration. Next slide, please. In terms of the models that we're using the CBOP model, or the Simplified Surface Energy Operational Model, which is a USGS data product available at global extent for various time steps from daily to annual. We're also collaborating with many different federal and university and government partners through the OpenET project, which is currently available in the Western continental United States for now, but there's plans to expand it. And this product takes in multiple models and formats, including the CBOT model. Here we see a sample output from the website, and this is also available via an API. And the CBOT model is available from this website and also from Earth Explorer, I believe. And feel free to reach out to Gabriel Sine, Meredith Reitz, or Toby Welburn for questions about this work. And next slide, please. Yeah, again, I'd like to thank all my co-authors and our many, many partners for their collaboration and for making this work possible. And feel free to reach out if you have any questions to any of the folks listed here. Thank you.
Thank you so much, Dr. Harlan. Next, we'll pass it off to Dr. Brian Pellerin to talk about the next generation water observing system.
All right, thank you. Today I'll be sharing some information on how the USGS is building a next generation water observing system for the United States. My name is Brian Pellerin. I'm the manager for the Next Generation Water Observing System program, which I'll also refer to as NGWAS. Next slide, please. The USGS operates the nation's largest water observing system, providing data that can drive forecasts of floods and droughts, guide management of reservoirs, and describe the current conditions and trends in water quantity and quality throughout the nation. This includes over 10,000 real-time stream gauges, more than 80,000 stream flow measurements per year, monitoring groundwater conditions at more than 17,000 wells, over 2,100 continuous water quality monitoring stations, 1.7 billion data requests in 2022 through our web services, and funding from both government appropriations and over 1,600 partners. Although this network has served the nation well, challenges of too much, too little, and poor quality water will become even more common in the future and require more data than the current networks can provide. Next slide, please. To address this challenge, the USGS began developing a next generation water observing system in 2018 to enhance the current water monitoring networks through the collection of high resolution data on water quantity, quality, and use. The NGWAS program also serves an important role as an incubator for the development and transition of new monitoring technologies and methods into national operations, and is supporting the modernization of our data telemetry, storage, and delivery systems for the USGS. Next slide. The enhanced monitoring is taking place in medium-sized basins, typically around 40,000 square kilometers, with the goal of providing data that can help improve water availability assessments, water management, and ultimately water prediction. 5 of those basins have been selected and are active. They're shown in white on the map and described in the bullets on the left side of the slide, with a goal of 10 basins for this pilot effort. Besides addressing national priorities, each basin has a focus topic that's addressed through new observing, research, and modeling. And just as an example, for the Upper Colorado River Basin, the focus is on understanding how snow accumulation and melt under a changing climate impact water availability for human and ecological uses. Next slide, please. The approach that NGWAS is taking within the basins is shown in this conceptual diagram. Some of those key activities include filling monitoring gaps in space and time, adding measurements of water budget components such as snow water equivalent, evapotranspiration, soil moisture, establishing sites for research and development of new technologies, improving the integration of remote sensing and in situ observations, and also improving the delivery of data to managers and to the public. Next slide. NGWAS is designed around a monitoring framework that integrates observations across space and time. For example, the intensive observations like those at research and development test beds, which we note at the top of this pyramid, are integrated with data that are more spatially dense across the landscape, but often at lower temporal frequencies, such as those from basin-wide monitoring surveys and satellite remote sensing. Nesting observations in this way across space and time allows us to better use the monitoring resources and learn as much as possible about water quantity, quality, and use within these basins. Next slide. Filling data gaps to improve the modeling and assessments is an important goal of NGWAS. For example, in this figure, you see the size of the water temperature network in the Delaware River Basin was approximately doubled as part of the NGWAS enhancements, which are shown as the colored circles on the map. This increase in the density of observations is guided by network design studies and achieved through both traditional methods as well as new approaches, such as the use of infrared cameras mounted on stream gauges and flown on drones. Next slide, please. NGWAS is also investing in monitoring that enhances the long-term data sets within the basins. This includes water budget monitoring, such as snow and soil moisture, synoptic surveys of water quality and quantity using autonomous vehicles, as well as manual sampling, geophysical data collection, and the use of cameras to capture imagery for both situational awareness and water science, as you can see in the graphic in this slide. Next slide. Investments through NGWAS and new technologies and methods, both within the USGS as well as with our university and industry partners, are leading to a number of advances around non-contact stream gauging, camera-based monitoring, autonomous sensing, and water quality monitoring technologies. And our goal is to transition as many of those capabilities as possible into our national operations within the next 3 to 5 years. Last slide. So in summary, the NGWAS program is leading the USGS toward a more modern future for water observing that will ultimately improve water science, management, and prediction through enhanced observations and new technologies. I'd welcome the opportunity to share more about our efforts. My contact information is listed on the slide, and I thank you for your time and attention.
Thank you so much, Dr. Pellerin. I'll turn it next to Dr. Laura Norman to talk about nature-based solutions to combat climate change with water.
Thank you. Greetings. My name is Laura Norman, and I am a scientist studying nature-based solutions that can be used to support and rehydrate drying streams of the North American Southwest. Next slide. Nature-based solutions use natural features or processes in the built environment to promote adaptation and resilience. Nature-based solutions are ecosystem-based approaches to achieve climate goals and combat the loss of nature. At COP27 last fall, the United States Biden-Harris administration released a roadmap for government agencies for nature-based solutions to address climate change, nature loss, and inequity. Beginning in 2004, the USGS, working with the U.S. Department of Housing and Urban Development, delineated colonias and the U.S.-Mexico border. Colonias are borderland neighborhoods that lack infrastructure and/or adequate housing. By mapping these boundaries and designating the colonia status, communities become eligible to receive specific funding to improve their quality of life. The U.S.-Mexico border is a hotspot for climate change, where warming incurs a loss of land and productivity, and marginalized communities like colonias are often subjected to impaired environmental health and not positioned well to adapt to perils associated with extreme drought or floods. Next slide. Natural infrastructure in dryland streams, or NIDS, are low-tech, low-cost structures made of rock, wood, or mud, installed by humans or beavers. One example is a gabion, a large rock detention structure wrapped in chicken wire, as pictured here. The International Boundary and Water Division contracted the USGS to apply watershed models to quantify the impacts of installing gabion NIDS as a nature-based solution in the colonias of Nogales, Sonora, Mexico, and Nogales, Arizona, to reduce the extremely hazardous and often deadly flooding there. As you can see in this example hydrograph, this natural infrastructure lowers peak flows and helps to negate overbank flows that can be hazardous. As a result, a series of gabions, NIDS, were installed around the perimeter of the watershed, and floodwater is now regulated downstream by the structures. Water is stored within the system for a longer period of time and then slowly released. Next slide. Just east of this, The U.S. Fish and Wildlife Service was working with Cuenca Los Ojos, a private NGO in Mexico, to restore the historic binational San Bernardino Cienega, a hotspot of biodiversity and treasured wetland habitat, using similar gabion-style NIDS. The USGS analyzed 30 years of Landsat imagery and documented a pronounced decline in vegetation correlated with steadily declining precipitation. However, at locations where natural infrastructure were installed, vegetation was maintained or increased despite our long-term drought conditions. This increased vegetation at NIDS was documented up to 5 kilometers downstream and 1 kilometer upstream of each structure. So NIDS help maintain and propagate plant growth with a very large spatial footprint and extend valuable growing seasons in arid land environments with a very large temporal footprint. Next slide. In the nearby U.S. Forest Service land, a private rancher installed over 2,000 smaller check dam NIDS throughout a small watershed. The USGS monitored the impact of these NIDS and documented the same decrease in peak flows with these structures that we had seen at the Nogales gabions, and also an increased volume of water being discharged over time by 28% compared to the control watershed. NIDS are a climate adaptation strategy that reduce flood hazards, but also harvest rainfall and augment water supplies over time, extending our precious seasonal water availability in arid environments and reducing impacts of drought conditions. Next slide. Using a calibrated watershed model, USGS estimated that 210 tons of sediment per year was stored behind this huge series of check dams in the Forest Service land, and found that the structures were maintaining function after 30 years of installation. So natural infrastructure in dryland streams, or NIDS, can regulate erosion by wind or water and increase soil stability and water quality. Likewise, beavers have long been recognized as agents of flood control and soil conservation, as referenced in this Idaho paper from 1950, parachuting in to install dams. But in the United States, the current population is only 10% of what it used to be. The reintroduction of beavers, sometimes coupled with the addition of beaver dam analogs, or BDAs, NIDS, can restore degraded wetlands, streams, and floodplains, and is becoming more popular. Beaver dams and their analogs are a cost-effective natural restoration tool that promotes drought and climate resiliency. This photo on the right shows a landscape scorched by fire, also in Idaho, where the beaver pond-infused riparian area and series of wetland ponds deflect wildfire spread and serve as natural refugia. Next slide. Working with scientists around the United States, we documented ways of soil carbon storage in all of these NIDS, including various size and construction of rock detention structures, beaver dams, and their analogs, to compare with wetlands, finding similar rates of sequestration due to NIDS' ability to capture soil, increase productivity and water availability, and store carbon underground. Rehydrating arid landscapes and conserving biodiversity using low-tech and low-cost natural infrastructure in dryland streams, NIDS, is something that community groups and advocates can do. This little animation shows pictures of different groups installing NIDS on the landscape. Community empowerment provides ownership of projects and a restoration stewardship economy. In our eco-region, we have established the Sky Island Restoration Collaborative to unite scientists, restoration practitioners, and land and water resource managers to improve their backyards and reverse cycles of degradation and climate change. Next slide. We have documented hundreds of examples confirming over and over again all of these climate adaptation and mitigation ecosystem services that NIDS can provide, given regular maintenance, proper site and scaling, and abundance of NIDS structures, well-trained practitioners to install them. The USGS uses satellite imagery, hydrologic modeling and monitoring, and technical innovation to document the impact of this low-cost, low-tech, nature-based solution and prioritize areas for their installations. It supports ecosystem services that protect communities by restoring water cycles that rehydrate arid watersheds and wetlands. Increasing vegetation increases moisture in the atmosphere, which ultimately cools our planet. This nature-based solution can reduce risk associated with climate change, nature loss, and inequity. Thank you.
Thank you so much, Dr. Norman. I'll now introduce our last presenter, Dr. Richter, to talk about environmental DNA.
Thank you very much for the invitation to present at World Water Week. I'm Kathy Richter. I'm a molecular biologist working for the U.S. Geological Survey, Columbia Environmental Research Center in Columbia, Missouri. I'm presenting today on innovations in biodiversity assessment and monitoring with environmental DNA. Next slide, please. So environmental DNA analysis is the identification of organisms from DNA sequences that are extracted from environmental samples. For example, water, soil, or air. I'll focus on water in this presentation. And in the photo, you can see me collecting a water sample. eDNA analysis is a complement to traditional survey methods, such as observations along transects, electrofishing, snorkel surveys, catch per unit effort studies, or mark recapture studies. eDNA identifications require knowledge of species' mitochondrial DNA sequences, but don't require taxonomic expertise to analyze each sample. This makes eDNA analysis a powerful tool to support management of biodiversity, including detecting rare species, defining species ranges, and detecting non-indigenous species. Detection rates and eDNA concentrations can be quantified, and this allows measurements of trends in diversity and in the abundance of individual populations. Next slide, please. Sampling to assess biodiversity with eDNA analysis is unique because eDNA analysis is very sensitive. A single copy of the target sequence can be detected in a quantitative PCR or digital PCR assay. This also means eDNA analyses are vulnerable to contamination and require careful technique and negative controls at each step. eDNA assays can be highly specific to detect only one target species and allow identification of cryptic species. Sampling for eDNA is generally non-invasive compared to traditional surveys because it does not require capture of target organisms and involves minimal disturbance of habitat. eDNA assays can be targeted to one species, with results analyzed by quantitative PCR or digital PCR, or assays can be taxa-specific, for example, targeted to all unionid mussels. And then the results can be analyzed with metabarcoding using next-generation sequencing. Next slide, please. eDNA analysis is a versatile tool for biomonitoring. Applications include monitoring populations, assessing effects of management actions. For example, the photo shows removal of invasive silver carp from Creve Coeur Lake in Missouri, and measurement of silver carp eDNA before and after the removal effort confirmed that most, but not all, of the silver carp were removed. eDNA monitoring can assist natural resource managers by informing decisions on responses to introduced species, actions to facilitate adaptation to anthropogenic changes, reintroductions of threatened species, and conservation introductions, which are now being considered in order to enhance ecological adaptation to climate change by moving species outside their historical native ranges in order to avoid extinctions or enhance ecosystem function. Next slide, please. One well-established application for eDNA monitoring is detection of aquatic invasive species. This slide presents a conceptual framework for early detection and rapid response, or EDRR. a coordinated set of actions to find and eradicate potential invasive species before they spread and cause harm. EDRR is considered a second line of defense after prevention measures. And eDNA analysis is useful at node B, surveillance and detection, and node F, evaluating response measures. Next slide. The ReadyNet project is under development at the USGS Ecosystems Mission Area and partner organizations, and is focused on early detection of aquatic invasive species with eDNA. ReadyNet stands for Rapid eDNA Assessment and Deployment Initiative and Network. So the goals are to test automated eDNA samplers and develop heuristics for when and where to deploy them, and often with the goal of deploying them in collaboration with the USGS Stream Gauge network. And develop a robust sample tracking system to track samples from collection through data analysis. We are also aiming to develop clear and consistent guidelines and standard operating procedures for eDNA analysis in order to increase reliability and build trust with natural resource managers. And at the center of the project is communication with decision makers, with stakeholders, among the scientific partners, and with the public. So at the completion of the project, the goal is an end-to-end toolbox that can be distributed across the United States and beyond, and be used by managers to protect critical resources from costly invasive species impacts. Importantly, these tools are not limited to aquatic invasive species. With the same tools or even the same samples, we can also use eDNA to monitor for important native species, for human health threats, and for fish and wildlife pathogens. Next slide, please. The standardization of methods and terminology for eDNA analysis is a global issue, and international partnerships are developing to advance the maturation and usability of this science. These examples are from Canada, the CSA Group, Fisheries and Oceans Canada, and the iTrack-DNA project, and from the United States, the Non-Indigenous Aquatic Species Database, and the ReadyNet project. Next slide, please. A large group of collaborators has recently come together to put forward a vision for a national eDNA strategy for the United States. And this was published this year in the Environmental DNA Journal. Many of the authors were participants in the second national workshop on marine eDNA, which was held in September of 2022. Figure 1 of the paper highlights potential applications of eDNA technology, including the detection of invasive species, monitoring of commercially important stocks, detection of pathogens and harmful algal blooms, and quantifying ranges and movements of native species. Next slide, please. And I'll end by thanking my colleagues who contributed to this talk. And here are some photos of automated eDNA samplers. And I'd also like to thank the USGS Ecosystems Mission Area. Thank you.
Thank you so much, Dr. Richter. And that is the end of all of the presentations. Thank you to all of our presenters today. I'm going to stop sharing my screen, and I think we can open the floor to some questions in the crowd. If you do have a question you would like to ask, please raise your hand or put it in the meeting chat, and we'll do our best to get to as many as we can. Looking at the chat right now, I have a question for Dr. Harlan. With regards to the triple approach, how good is compatibility between different approaches?
Thank you for that question. I briefly typed something in the chat, but The answer is, unfortunately, it does depend on, where you're looking for that validation. I did link the paper to the CBOT model that I highlighted, and it shows a range of correlation values between 0.87, so really capturing that monthly flux tower well. And then in some regions, it's not performing quite as well, with the lowest outlier being 0.09. And so I think this work is still in development, and those were results with MODIS, and perhaps with Landsat we can increase accuracy. So I encourage you to check out that paper, and Chris might also have other resources for validation of the USGS evapotranspiration product. Yeah, thank you for that question.
Thank you. Chris, anything to add?
Yes, sorry, I was getting unmuted and visualized here. Yeah, so I would fully agree with Merritt. I think the one thing that we're doing is just simply providing, through the model that Merritt has talked about, we're providing that data globally on demand. It's generally considered provisional for the points that Merritt just highlighted, but you can get that data anywhere. And so what we like to do with our on-demand data products, these products are considered to be on the path towards an operational readiness state. So being able to, for the greater global community to get access to these products and give us feedback, it enables us to produce a higher quality product at a future time. So I did provide the link. That paper's good, and the link directly to the data for your location, if you're interested, is provided there in the chat. Thanks.
Thank you, Chris. We have another question for Dr. Richter. What is the status of genomic libraries for comparison with eDNA data, in particular with regards to US-wide application and different ecosystems?
Yes, thank you for this great question. This is an important limitation of eDNA analysis in that we can only identify species by DNA sequence if the DNA sequence is known, at least for the barcode region of the mitochondrial DNA, generally the cytochrome oxidase 1 gene. There are databases that are growing for these sequences. The National Center for Biotechnology Information in the United States hosts the GenBank database, and also there is the Barcode of Life database, which collects this information. And so as these databases grow, the power of eDNA analysis will increase. And right now, coverage of vertebrate species is quite good, but coverage of invertebrate species is sometimes lacking. Thank you.
Thank you so much, Dr. Richter. Next question we have, how accurate is the available data on groundwater availability and rate of withdrawal? I'm guessing this is for NGWAS, but we'll open it to the entire panel.
Thanks. Perhaps this is related to some of the remote sensing observations, but I'll weigh in on the NGWAS perspective. I mean, we are doing a lot of on-the-ground monitoring, so certainly where we're making measurements in groundwater wells, the accuracy is very high. I think from the perspective of a national network, a bigger challenge might be that we may not always have the spatial distribution of sites that are necessary to develop a clear understanding of groundwater availability and withdrawal at larger scales, in particular regional to national scales. But I think if perhaps there's a response related to remote sensing applications specifically, maybe Merritt or Chris could comment on that.
Merritt, Chris, do you have anything to add?
I think Brian summarized it pretty well. I'm aware of some, groundwater modeling from remote sensing through MODIS, for example, but that's at much kind of coarser levels. And so it's really important to have these on the ground, really high resolution measurements to help balance that out. Maybe Chris has something more to add though.
Yeah, so the observations for groundwater typically come from the active domain. So there's the satellite called SMAP, NASA's Soil Moisture Active Passive satellite. So the USGS, in terms of remote sensing, are not actually making those observations, but we're relying on NASA to make those, to actually acquire those observations. But that would be my recommendation, is to explore the active domain of remote sensing for the best groundwater retrievals. Thanks.
Thank you, everyone. Our next question is to Dr. Norman. The effects of nature-based solutions in water can be hard to isolate from other impacts on the basin, climate change, outside factors, etc. Did USGS have or find techniques in monitoring the effects that specifically benefit from nature-based solutions?
Thank you, and I apologize, my camera is being funny today, but I agree that's very difficult to do. And in my research, we attempt to acquire baseline studies to quantify the water budget with no disturbance or human impacts to try to analyze this. And using satellite imagery, as described, we've been able to step back through time to see this, so establish those baseline conditions. And in addition, we develop before and after impact into our experimental design using paired watersheds, thereby teasing out the expected response to climatic events using the control as a baseline, and then compare that to a treated site that has natural infrastructure installed.
Thank you, Dr. Norman. Do we have additional questions from the crowd attendees? Please put them in the chat, and I'll read them out here. Last call. If there are no other questions, I think we may wrap early. So thank you so much. Oh, yeah. Annalise, Dr. Annalise Bloom, would you like to provide some closing statements for the session?
Yes. Okay. Sorry. Having trouble for some reason. I clicked video and mute and it was freezing on me. Thanks so much, Joe. And thanks so much to all of these presenters. Really appreciate the interesting highlights of their work from satellites to eDNA that are helping us better understand water resources both in the United States and around the world. Thanks so much to all of you for attending, for your time and interest in this session. I encourage you to reach out to the panelists if you're interested in learning more about their research and science. And really our hope for this session was that it leads to future conversations and collaborations that can help us truly unleash the science and take advantage of all of the amazing scientific advances being made by the USGS and scientists more broadly to help us understand our changing world and inform policy and decision-making. So thanks so much again. Really appreciate your attention. This concludes the session.